SwayamPrabha. Digital India. Educated India.
Welcome to Proteomics course. Today we will talk about enzymes, its basic concept and catalytic and regulatory strategies. So, lecture outline is that first we will talk about different basic concepts of enzyme, including enzyme kinetics, energetic and enzyme inhibition. We will then move on to different type of catalytic strategies and regulatory strategies. So, as you all know, enzymes play a very important role in biochemistry and offer sensitive and specific method of quantitation for various substances. All enzymes are proteins. Therefore, I think it is essential to study about enzymes while studying the basic concepts of amino acids and proteins, although it may not be directly linked to the proteomics, but understanding about proteins and enzymes is very fundamental for the advanced understanding of concepts related to the proteomics. So, what are enzymes? Enzymes are molecular catalysts. Almost all the known enzymes are proteins, and as we have discussed in the previous lecture, there are 20 amino acids which offer array of chemical forces. These enzymes can accelerate the given reaction up to even million folds. For example, let us take example of an enzyme carbonic anhydrase, which catalyzes hydration of carbon dioxide. Now, this enzyme can catalyze 10 to the power 6 molecules per second. These enzymes are highly specific and they catalyze single or very closely related reactions. There are various examples which we will talk during the entire lecture, how enzymes regulate various reactions. There are certain proteolytic enzymes, for example, which catalyze proteolysis and hydrolysis of peptide bond. In terms of enzyme specificity, let me give you a few examples. Trypsin, it is highly specific. It cleaves bond only on the carboxylic side chain on the residues lysine and arginine. Another enzyme, thrombin, which participates in blood clotting. It is even more specific than trypsin. It catalyzes hydrolysis of arginine glycine bonds in a very specific peptide sequence and therefore offers highly specificity. There are 6 major classes of enzymes which are grouped according to the type of reactions they catalyze. I will not go into too much detail, but briefly let me talk what type of reaction these class of enzymes are offering. Oxidoreductase, it catalyzes oxidation and reduction. Example is pyruvate dehydrogenase. Transferase, these enzymes can transfer the certain groups. So, group transfer can be mediated by this group of enzymes. Example, hexokinase. Hydrolase, these can catalyze hydrolysis reaction. Example, chymotrypsin. Lyase is another group of enzyme which adds group to double bonds or remove groups to form the double bonds. Example include fumarase. Isomerase, as the term says, isomerization. So, these group of enzymes can transfer groups within the molecule. Example, phosphoglucoisomerase. Ligase, it catalyzes bond formation by using ATP. Example, glutamine synthetase. These are just few examples, but each enzyme class incorporate various enzymes. Broadly, these are the type of reaction which these enzymes can catalyze. So, let us talk about enzyme activity and how various cofactors govern these enzyme activity. So, many enzymes catalytic activity depends on the presence of small molecules which are known as cofactor. There could be two groups here as shown in the PPT, metals and small organic molecules. The coenzymes are a small organic molecule such as those derived from the vitamins. The prosthetic group, that is a tightly bound coenzyme. The apoenzymes are enzyme without its cofactor, and holoenzyme when you combine cofactor and apoenzyme to give rise to complete catalytic enzyme, that is holoenzyme. Most enzymes are made up of a protein part known as the apoenzyme as well as a cofactor which can either be an organic molecule known as coenzyme or a metal ion. These cofactors are essential for the enzyme to be catalytically functional, and the complete functional enzyme is referred as holoenzyme. The pyruvate dehydrogenase is a complex enzyme which uses thiamine pyrophosphatase as its coenzyme, while another enzyme, carbonic anhydrase, uses zinc ion as its cofactor. Enzymes are classified on the basis of the reactions that they catalyze. Most enzymes are named by adding the suffix ase either to their substrate or the type of activity they carry out. However, as more enzymes came to be known, it became increasingly difficult to name in this manner. Classification by international organizations has therefore led to 6 enzyme classes with many subgroups within each class, depending upon the type of reactions they are catalyzing. Every enzyme has a unique 4-part classification number known as the enzyme commission number or EC number, in which subclass number gives finer detail about that particular enzyme reaction. Let us now talk about free energy and enzyme. So, remember the concept of entropy, delta G equals to delta H minus T delta S. So, delta G must be negative for a spontaneous reaction. So, the energy which is required to convert the substances to product, that is rate of reaction, and enzymes can only affect the rate of a reaction. So, enzymes cannot alter reaction equilibrium, they can accelerate attainment of equilibrium concentration. So, what is transition state and activation energy? When you take a reaction from substrate to product, it goes through intermediate transition state. The activation energy is differences in free energy between transition state and substrate. The activation energy is a barrier to reactions. Now, if you look at this graph, enzyme decreases the activation energy and alter the rate of attainment of reaction equilibrium. by facilitating the formation of S star. Conversion of substrate to product proceeds through formation of a transition state. The free energy of activation of an uncatalyzed reaction is very high. Enzymes form favorable interactions with the substrate and facilitate formation of the transition state by lowering the free energy of activation. The transition state then dissociates to give the product and regenerates free enzyme. For a reaction to be spontaneous, the delta G must be negative. It must be emphasized that enzymes do not alter the equilibrium of a reaction. Enzyme substrate complex. The enzyme substrate complex or ES formation is first step in catalysis. Enzymes are very selective in choice of substrate they can bind, which ultimately dictates the specificity of these catalysts. These substrates are bound to active site of an enzyme. There have been experimental evidences that enzyme substrate or EX complex do form, and various strategies which have shown that ES complex forms include maximal velocity, X-ray crystallography and spectroscopy. So, what is active site? The active site is a region of enzyme that binds to substrate. It provides a three-dimensional cleft. It is formed by amino acid from different parts of the sequence. So, it is not necessarily that the residues in that pocket are only going to form the active site, but even it can be from different regions of given protein which could form the active site. They occupy only small portion of enzymes, and some extra amino acids they help in scaffolding. To understand enzyme substrate binding, different models have been proposed. The two popular models, one is lock and key model, which was proposed by scientist Emil Fischer in 1890. This model explains the complementary interaction can determine the specificity. So, if you have an enzyme and a substrate is present, now the active site of unbound enzyme should be complementary in shape to the substrate, something similar to a key is very specific for a given lock, then only it will open. Other model is known as induced fit, which was proposed by scientist Daniel Koechlin in 1958. So, the active sites they form a shape which is complementary to the substrate and only after the substrate has been bound. So, enzyme can change the shape of a substrate binding. In this case, the substrate structure do not have to be specific to the active site of enzymes. It can be dictated by enzyme and the shape can be changed, which will be complementary for this binding. Fischer's hypothesis has defined as the lock and key hypothesis. Any lock which is analogous to an enzyme can have only one suitable key of appropriate shape and size to open it. The various available keys which are analogous to the thousands of substrates available can attempt to open the lock, but only one will be the perfect fit that can open the lock. Similarly, only one particular substrate will fit into the active site of the enzyme and the enzymatic reactions can occur. According to the Fischer's hypothesis, enzymes and their substrates possess a specific complementary geometric shapes that fit exactly into each other. This model accounts for the specificity of enzymes that fail to account for stabilization of the transition state. Koshland proposed another hypothesis known as Koshland's modified hypothesis, suggested that the active site of an enzyme gets continually reformed based on the interaction that it establishes with the substrate molecule. This hypothesis accounts for both the enzyme specificity and the stabilization of the transition state, since enzyme is not considered to be a rigid molecule. Let us talk about enzyme kinetics. In 1913, scientists Michaelis and Minton, they derived a model to explain the concept of enzyme substrate complex, which is fundamental to understand the enzyme reactions. The Michaelis-Menten explained kinetic characteristics. The role of enzyme catalysis or V0, which is number of moles of product formed per second. It varies with substrate concentration S. It increases linearly as the substrate concentration increases, levels off at a maximal velocity at higher substrate concentration. So, an enzyme E which catalyzes a substrate S to give rise to a product P can be understood in this equation: E plus S giving rise to E S. An enzyme substrate complex is breaking down to enzyme plus product. Now, E combines with S to form E S complex in presence of rate constant K 1, whereas the E S complex can be dissociated to form E N S with rate constant K minus 1. E S complex can form product with rate constant K 2, and E S complex can be reformed by reverse reaction in rate constant K minus 2. So, here is Michaelis-Menten equation. The rate V 0 of formation of product can be derived by this equation: V 0 equals to V max substrate concentration S divided by S plus K m, where K m is Michaelis constant. Now, this equation is fundamental to understand various problems. So, let us say we take this equation and there is one situation where substrate concentration is less than K m or Michaelis constant. Now, what will be the equation? In this case, V 0 will become V max divided by K m times substrate concentration. So, rate is directly proportional to substrate, which is the reaction is first order. Now, let us take another scenario where substrate concentration is greater than K m. Now, what will be the equation? V 0 equals to V max. So, rate is maximal and it is independent of substrate concentration. In this case, reaction is of 0 order. Now, let us take third situation where substrate concentration is equal to K m. Now, in this case, equation will become V 0 equals to V max divided by 2. So, substrate concentration at half maximal velocity is known as Michaelis constant or K m. So, K m is substrate concentration at half maximal velocity. As you can see in the graph, the K m is a measure of strength of enzyme substrate complex. So, higher K m indicates weaker bonding and low K m is strong binding. So, another scientist gave Lin-Vivier-Burke plot or double reciprocal plot, which is a plot of 1 by V 0 versus 1 by S. It is reverse of the V 0 or S, which was taken in case of Michaelis reaction. Enzymes catalyze the formation of product from its substrate via an enzyme substrate intermediate complex. During the initial stage of the reaction, the equilibrium favors product formation rather than dissociation of the ES complex to give back the substrate molecules. The number of moles of product formed per second during these stages determines the reaction velocity for that particular enzyme. V 0 has an almost linear relation with substrate concentration. When the substrate concentration is low, it becomes independent at higher concentrations. The Michaelis-Menten model for enzyme kinetics assumes that the breakdown of ES complex to give back free substrate is negligible, and also assumes steady state conditions whereby the rate of formation and breakdown of enzyme substrate complex are equal. The reaction velocity increases linearly with substrate concentration when S is low, but becomes independent at higher concentrations. The maximal velocity V max that can be achieved by an enzyme refers to the state in which all its catalytic sites are occupied. The substrate concentration at which the reaction velocity is equal to half its maximum value is known as Michaelis constant Km. The Lin-Vivier-Burke equation or the double reciprocal plot is a useful tool that can be plotted using simple experimental data from kinetics experiments. This equation is derived from the Michaelis-Menten equation by taking reciprocals on both sides and then plotting a graph of 1 by V 0 versus 1 by substrate. The y-intercept on this graph can be used to deduce the value of V max, while the x-intercept gives the value of Km. Enzyme inhibition. The enzyme inhibition can provide insights into catalysis. Enzyme activity can be inhibited by small molecules and ions. The enzyme inhibition provides major control mechanism in biological systems. There are two types of enzyme inhibition: reversible and irreversible. The reversible inhibitors, they dissociate rapidly, and the enzyme inhibitor EI complex is dissociated. There are three different strategies for reversible enzyme inhibition: competitive, non-competitive, uncompetitive or mixed inhibitions. So, let us discuss first situation, competitive inhibition. The enzyme binds to substrate or inhibitor and forms enzyme substrate or enzyme inhibitor complex, but do not form enzyme substrate inhibitor ESI complex. The inhibitor is similar in structure to the substrate, which binds the active site of enzyme. It can reduce the rate of catalysis and also reduces the number of enzyme substrates which are formed by binding at the same active site as shown in the figure. Uncompetitive inhibition. Inhibitor and substrate, they bind at different sites. Enzyme substrate inhibitor complex is formed. So, by increasing the substrate concentration, inhibition cannot be overcome, which was the case in the previous one of competitive inhibition. The reaction rate increases more slowly at lower substrate concentration as compared to the uncompetitive. After talking to reversible enzyme inhibition, now let us discuss irreversible enzyme inhibition. Inhibitor is very tightly bound to enzyme either covalently or non-covalently in such a way that this inhibition is irreversible. Let us take the very classical example, penicillin, the first antibiotic which was discovered. It was irreversibly inactivating a key enzyme, transpeptidase, in bacterial cell wall synthesis and killed the bacteria. Now, in irreversible enzyme inhibition, there are different classes: group specific, reactive substrates, or suicide inhibitor. The group specific irreversible enzyme, these inhibitors can react with the specific side chain of amino acids. The reactive substrates or affinity labels, they are structurally similar to the substrate and covalently bind to the active site. They are more specific for enzyme active site than group specific reagents. The suicide inhibitors, they are highly specific substrates which can modify the active site of an enzyme. They are most specific for modification of enzyme's active site. Enzyme inhibition can either be reversible, where the inhibitor can dissociate quickly from the enzyme, or irreversible, where the inhibitor dissociates very slowly from the enzyme and can covalently modify the enzyme, thereby rendering it unsuitable for further catalytic reactions. The reversible inhibition can be further classified as competitive, uncompetitive, and mixed inhibition. In competitive inhibition, the inhibitor molecule is structurally similar to the substrate and therefore binds to the enzyme at the active site. Binding of inhibitor prevents substrate from binding, thereby decreasing the reaction rate. The Vmax in this type of inhibition remains the same and only Km is altered. The competitive inhibition can be overcome by suitably increasing the substrate concentration, which allows the substrate to outcompete the inhibitor for the enzyme's active site. In case of uncompetitive inhibition, the substrate and inhibitor both have different binding sites on the enzyme. Binding of inhibitor to the enzyme substrate complex prevents any further reaction and no product formation is observed. Both the Km and Vmax are found to decrease with this type of inhibition. Mixed inhibition or non-competitive also binds to the enzyme at a site distinct from the substrate binding site. However, the difference is that it can bind either to the enzyme or enzyme substrate complex. Binding of either one brings about conformational changes in the enzyme structure, thereby affecting binding of the other. This inhibition can be reduced, but not overcome by increasing substrate concentration. Both Km and Vmax are altered in this type of inhibition. Now, for discussing various basic concepts of enzyme, let us move on to catalytic strategies. Enzymes are very effective and extremely specific catalysts. The enzymes employ multiple mechanisms to facilitate catalysis. For example, covalent catalysis, acid base catalysis and metal ion catalysis. In the next few slides, we will talk more specifically about these strategies. So, covalent catalysis. In this, the active site contains reactive group which covalently attaches to the substrate. Let us take example of chymotrypsin, which is contain a highly reactive serine at the position serine 195. It plays a very central role in catalytic activity of chymotrypsin. Covalent catalysis involves the formation of a transient covalent bond between the nucleophile present in the enzyme and the substrate molecule. Formation of this bond provides an alternative reaction pathway that has lower activation energy than the uncatalyzed reaction. Several amino acid side chains act as effective nucleophile that facilitate this reaction. The enzyme is regenerated in its unaltered form at the end of this reaction. Chymotrypsin is one such enzyme that carries out catalysis by covalent modification. It possesses a catalytic triad of histidine, aspartic acid and serine at its active site, with the serine at position 195 serving as highly powerful nucleophile. The reaction between the serine hydroxyl group and unreactive carbonyl group of the substrate helps in bringing about product formation. Enzyme regeneration occurs after this reaction. Covalent modification of the serine residue led to the irreversible inactivation of the enzyme. Acid base catalysis. A molecule plays a role of a proton donor or acceptor. This is other than the water molecule. Let us take example of carbonic anhydrase, where zinc ion is bound to imidazole ring of 3 histidine residues and water molecule. So, histidine facilitates hydrogen ion removal from zinc bound water and generates hydroxyl ion. Biochemical reactions involving the formation of unstable charge intermediates are often stabilized by transfer of protons to or from the substrate or intermediate. For non-enzymatic reactions, acid base catalysis may involve only the hydronium or hydroxyl ions of water, referred to as specific acid base catalysis. In many cases, however, water alone does not suffice to catalyze the reaction. In such cases, proton transfer is facilitated by weak organic acids or bases. The organic acids act as proton donors, while organic bases can serve as proton acceptors. In case of enzyme catalyzed reactions, weak proton donors or acceptors are often present as amino acid side chain at the active site of enzyme itself. The precise positioning of these groups within the active site mediate proton transfer reactions, which can provide rate enhancement of several orders of magnitude. The acid base catalysis is a common mechanism of action employed by many enzymes. It is often used in combination with another mechanism such as covalent catalysis. The ease of stabilization of charged intermediates by the amino acid side chain helps in lowering the activation energy for product formation. Chymotrypsin is one such enzyme that employs both covalent catalysis as well as acid base mechanism. The arrangement of the catalytic triad consisting of aspartic acid, histidine and serine at the enzyme's active site is such that the histidine residue serves as a general base catalyst by polarizing the hydroxyl group of serine. The alkoxide ion thus generated in the serine residue makes it a powerful nucleophile. Following substrate binding and nucleophile attack of the serine on the carbonyl group, the geometry of intermediate becomes tetrahedral and the negative charge developed on the carbonyl oxygen gets stabilized through interaction with other side chain of the proteins in a site known as oxyanion hole. An internal proton transfer then causes the tetrahedral intermediate to collapse and generate acyl enzyme intermediate, after which amine group is released from the active site. Once the amine group leaves the enzyme's active site, it is replaced by a molecule of water, which carries out hydrolysis of ester group of acyl enzyme intermediate. Mechanism for hydrolysis proceeds via formation of a tetrahedral intermediate with histidine acting as a general acid catalyst and the negative charge on oxygen being stabilized by residues in oxyanion hole. The tetrahedral intermediate then breaks down to liberate the second product in the form of a carboxylic acid along with regeneration of enzyme, which is then ready for another round of catalysis. Internal proton transfers between amino acid side chains therefore play a vital role in acid base catalysis by enzymes. Third strategy is metal ion catalysis. The metal ions serve as bridge between enzyme and substrate. They increase binding energy and hold substrate in appropriate conformation which is required for catalysis. Let us take example of nucleoside monophosphate kinase NMP. So, all these enzymes utilize ATP as substrate. The NMP kinase catalyze interconversion of ATP and NMP into nucleoside diphosphate NDP by transferring the phosphoryl group. Metal ions either present in solution or bound to the enzyme itself facilitates catalysis by forming favorable interactions between enzyme and substrate or in the transition state. Metal ions in the active site of enzyme typically react with a water molecule and activate it by facilitating generation of a strong nucleophile in the form of a hydroxide ion. The nucleophilic alkoxide ion attacks the unreactive carbonyl group to form a tetrahedral intermediate. The tetrahedral intermediate, in which the charges are stabilized by the metal ion, these favorable interactions help in orienting the substrate and enzyme in suitable position for transition state and subsequently product formation. Carbonic anhydrase is an enzyme responsible for hydration and dehydration reactions of carbon dioxide and bicarbonate respectively, and has been found to have a divalent zinc ion associated with its activity. The zinc ion in its active site is bound to the imidazole ring of 3 histidine residues as well as to a molecule of water. This binding to water facilitates formation of hydroxide nucleophile with concomitant release of a proton. The generated hydroxide ion at the active site then attacks the carbon dioxide substrate, converting it into a bicarbonate ion. The negative charge generated on the oxygen atom is stabilized by interaction with the zinc ions. Binding of another molecule of water to the zinc ion at the active site of enzyme leads to release of the bicarbonate ion and regeneration of the enzyme molecule for another round of catalysis. Various pH related studies has provided substantial proof for this mechanism. After briefly discussing the catalytic strategies, now let us talk about regulatory strategies. The metabolic pathways are very complex, especially when we are talking at the proteome scale. So, it includes one or more enzymes which exhibits greater effect on rate of overall sequence. So, in response to a given trigger or a signal, these regulatory strategies of enzymes can modulate catalytic activity and allow cells to meet the energy requirements, and eventually it will dictate the cell growth and survival. The first strategy is allosteric regulation. The allosteric proteins possess regulatory sites and multiple functional sites. The activity is modulated by specific regulatory molecules. These regulatory molecules bind at different site than the active site and trigger conformational changes which are transmitted to the active site. The allosteric proteins possess properties of cooperativity. So, activity at one functional site can affect the activity of others in cooperative manner. So, the allosteric enzymes are information transducer. In response to the signal molecules, their activity is modified. Let us take an example hemoglobin, where oxygen binding curve is sigmoidal, that is a cooperative binding behavior. So, binding of hydrogen ion and carbon dioxide promotes release of oxygen, and there is allosteric linkage between binding of hydrogen ions, carbon dioxide and oxygen. Activity of all enzymes must be regulated to ensure that they function only to the desired extent at the appropriate locations within an organism. Common mechanism of regulation include allosteric or feedback inhibition, control of isozyme forms, reversible covalent modifications and proteolytic activation. Allosteric enzymes. These enzymes possess distinct regulatory and catalytic sites, and these are often found as the first enzyme of a reaction pathway. Regulation of the first enzyme of a pathway by the final product of pathway is known as feedback inhibition. Binding of regulator molecule to the regulatory site of the enzyme triggers a series of conformational changes that are ultimately transmitted to the active site, where substrate binding is then inhibited. It has been observed that allosteric enzymes do not obey regular Michaelis-Menten kinetics. Aspartate transcarbamylase, which catalyzes the first step of pyrimidine biosynthesis, is an allosteric enzyme having distinct regulatory and catalytic subunits. The binding of substrate to the catalytic subunits induces conformational changes that stabilize the relaxed state or R state of the enzyme, thereby facilitating the enzymatic reactions. The inhibitor for this enzyme is CTP, which is the final product of the pathway. Binding of inhibitor to the regulatory subunits stabilizes the tense state or T state of the enzyme, thereby preventing the reaction from taking place. Isozymes or isoenzymes are multiple forms of enzymes. The isozymes catalyze the same reaction, but differ in structural characteristics. Isozymes varies regulation of same reaction at distinct locations. They differ in Km value, Vmax and different regulatory properties. Isozymes are expressed in different organelles or even at different stages of development. So, essentially it can allow the very intricate development of metabolism by fine tuning various reactions responsible in metabolism to meet the need of a given tissue or developmental stage. Isoenzymes are homologous enzymes within a single organism that differ slightly in their amino acid sequence, but catalyze the same reaction. These enzymes are mostly expressed in different tissue and have different kinetic parameters such as substrate affinity Km and maximum velocity Vmax. Lactate dehydrogenase is an enzyme involved in anaerobic glucose metabolism that is present as 2 isozyme forms in human being. The tetrameric HALT enzyme, which requires an aerobic environment to function, has higher affinity for its substrate than the muscle enzyme. Despite having 75% of sequence homology, they also differ in that high levels of pyruvate allosterically inhibit the heart enzyme, but not the muscle form. Now, let us talk about reversible covalent modification. This is a very effective method to control enzyme activity. You have heard the example of phosphorylation. So, protein kinases, they can catalyze the phosphorylation reactions, and ATP is served as phosphoryl donor. There is another enzyme, protein phosphatase, which removes the phosphoryl group by hydrolysis process. Reversible covalent modification is another commonly employed enzyme regulatory strategy. This is most widely observed modification is phosphorylation, which is carried out by various enzyme kinases with the help of ATP as a phosphoryl donor. Some enzymes are more active in their phosphorylated forms, while others are less active in this form. Dephosphorylation is carried out by the phosphatase enzyme. Apart from phosphorylation, which most commonly takes place at serine, threonine and tyrosine residues, other reversible covalent modifications include adenylation, uridylation, methylation and ATP ribosylation, which modify different amino acid residues of the protein. Now, last regulatory strategy is enzyme activation by proteolytic cleavage. So, enzymes can be controlled by this mechanisms which cycles between its active and inactive states. The inactive state, which is precursor, is known as zymogen or proenzyme. One of the classical example is the enzyme chymotrypsin, which is present in the digestive tract. So, trypsin converts chymotrypsin, which is a zymogen, into the active chymotrypsin by hydrolyzing peptide bond. The blood clotting due to the cascade of zymogen activation is another example. Several enzymes exist in their inactive forms known as zymogens, where they do not possess any catalytic activity. In order to become active, they need to be activated by hydrolysis of one or more peptide bonds by various proteases. The removal of certain regulatory residues irreversibly converts the enzyme into its active form. Unlike reversible modification, the enzyme is degraded after completion of catalysis. Several digestive enzymes as well as clotting factors are regulated by proteolytic activation. Chymotrypsin, a digestive enzyme that hydrolyzes protein in the small intestine, exists in its zymogen form within membrane bound granules after synthesis in the acinar cells of pancreas. The proteolytic enzyme trypsin converts it into its fully active form by cleavage of a peptide bond between arginine at position 15 and isoleucine at position 16. The resulting enzyme known as pi-chymotrypsin is acted upon by other such molecules to yield the completely active and stable alpha chymotrypsin, which consists of 3 chains linked by interchain disulfide bonds. So, in summary, today we discussed various concepts related to enzymes which are highly effective catalysts. There are several catalytic mechanisms which are employed by the enzymes, which include acid base catalysis, covalent catalysis and metal ion catalysis. We discussed some of these mechanisms in some detail. The activities of metabolic pathways in cells are regulated by control of the activities of certain regulatory enzymes, and 4 different regulatory strategies were discussed. So, I hope now you have some basic understanding of enzymes and different catalytic and regulatory strategies. Thank you.